Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Izzuddin bin Abdul Razak, and today I'm gonna present about chili peppers which enhance resistance to bacterial spot disease. Chili peppers are an important crop around the world, valued for their pungent fruit and culinary versatility. However, chili plants are susceptible in various diseases, including the devastating bacterial spot caused by the bacterium Xanthomonas. The application of traditional control methods, such as the use of copper-based bacteria, has limited effectiveness and can have a negative impact on the environment. The widespread use of copper-based bacteria has also led to the development of copper-resistant strains of Xanthomonas, further complicating disease management. To address this challenge, researchers have explored the potential of genetic engineering to develop chili pepper varieties with enhanced resistance to bacterial spot disease. BS2 resistance gene from wild chili peppers is the gene used to produce transgenic crop. BS2 resistant gene source from wild chili peppers has emerged as a valuable asset in combating bacterial spot disease caused by Xanthomonas in transgenic chili plants. This gene encodes an immune receptor that can identify specific effector proteins released by Xanthomonas bacteria, prompting a defense mechanism within the plant. Studies have revealed that transgenic chili peppers line expressing the BS2 gene exhibit notably reduced disease symptoms and increased yield compared to non-transgenic counterparts were exposed to Xanthomonas in field trials. Method of transgenic approach used for gene transfer. The first one is agrobacterium-mediated genetic transformation. This method involves the use of Agrobacterium tumefaciens, a natural genetic engineer, to transfer the BS2 gene into the target plant cells. Agrobacterium carries a tumor-inducing plasmid, TI plasmid, that integrates a segment of its DNA, including the BS2 gene, into the plant genome. The transformed plant cells then develop into transgenic plant expresses the desired resistant gene. The second one is plant tissue culture. Prior to gene transfer, plant tissue culture techniques are often employed to regenerate whole plants from isolated cell or tissues. This process allows for the efficient transformation of a large number of plant cells within the BS2 gene, ensuring successful integration and expression of the resistant trait in the resulting transgenic plant. The third one is selectable marker genes. In conjunction with the BS2 gene, selectable marker genes are often included in the genetic transfer process to aid in the identification and selection of successfully transformed plant cells. These marker genes confer resistance to antibiotic or herbicides, allowing researchers to distinguish transformed cell from non-transformed ones during the regeneration process. The last one is molecular characterization. Following gene transfer, molecular techniques such as polymerase chain reaction or PCR and sequencing are used to confirm the presence and expression of the BS2 gene in transgenic plant. This method ensures the stable integration of the resistant gene and its functionality in conferring protection against bacteria. For the result of the application, it effective resistant against bacterial spot disease. Its broad spectrum protection. The introduce of BS2 resistance was effective against diverse strains of Xanthomonas, indicating its potential for providing broad spectrum protection. This The third one is improved crop yields. Field trials demonstrate that the presence of the BS2 gene in susceptible peppers varieties reduce disease to extremely low levels, with yield or marketable fruit typically 2.5 times higher than not transformed parent line. Next is potential for durable resistance. The involvement of the bacterial virulence gene AVRBS2 in pathogen fitness and its prevalence in many Xanthomonas suggests that the BS2 gene may be durable in the field. And the last one is successful gene transfer. Agrobacterium mediated genetic transformation has been used to successfully transfer the BS2 gene from wild chili peppers into commercial cultivars of chili plants. In conclusion, the application of the BS2 resistant gene from wild chili peppers has been a big win for protecting chili crops from bacterial spot disease caused by Xanthomonas bacteria. By adding this gene into the commercial chili varieties through genetic modification with Agrobacterium, we have seen a significant reduction in disease symptoms and increase in yields during field trials. The BS2 gene offers strong and lasting protection against a variety of pathogen strains, making it a valuable tool for sustainable chili pepper farming. This research confirmed that incorporating the BS2 gene into crops through genetic engineering is the practical and effective way to boost crop resilience and productivity against bacterial spot disease. That's all from me. Thank you.